Hello everyone, welcome back to my channel Competitive Botany. In this video we are going to see recombination mapping with a 3-point test grass. In the previous video we are already discussed about 2-point test grass. In this video we are going to discuss a 3-point test grass. So if we take 3 genes at a time to observe their recombination frequencies, then it is called as 3-point test grass. Okay, so if we take 3 genes at a time, okay, so if we take 3 genes to observe their recombination frequencies, that is called 3-point test grass. It is generally used to measure the distance between 3 genes present on single chromosome. Okay, so it is used to measure the distance between 3 genes. That's why this is called as 3-point test grass. Okay, so Bridges, the scientist Bridges, when he was working on Drosophila, he crossed a wild variety with a mutant Drosophila. He observed 3 X-linked genes. Okay, so they are for scoot bristles, echinus eyes, cross veinless wings. So these are the 3 different characters. So they are governed by 3 different genes. So in wild varieties, they are designated by SC plus, EC plus, CV plus. In the case of mutant variety, they are denoted as SC, EC, CV with the small letters. So now we are going to make a cross in between a wild and mutant varieties. Now we are going to see the distance between these 3 genes. Okay, that's why this cross is called as 3-point test cross. Let's see. So he has made a cross in between mutant female with a wild male. Okay, so now we are going to get F1 progeny. Before that, from this one gamete will come and from this one gamete will come. So in future they are going to meet each other, resulting in F1 hybrid. Let's see. So these are the gametes produced from both individuals. So they are going to meet each other, resulting in the formation of F1 hybrid. So this is the F1 hybrid. So it receives one chromosome from female parent and one chromosome from male parent. So it's a hybrid. Okay, so it is going to cross with a double recessive parent. Then the cross is called as test cross. So now we are going to do a test cross. Okay, so here when the F1 hybrid is backcrossed with the mutant parent, so then we are going to get the progeny. So the progeny, before going to get the progeny, there are chances of recombination in between these 2 chromosomes. Okay, so there is a first chance to occur recombination in between these 2 genes, SC and EC genes. That is the first chance. There may be second chance to occur a single crossover in between these 2 genes. There is a third chance to occur double crossovers in between SC and EC and CV. So this is the double crossover. Okay, you need to remember. Depending upon this, we are going to get the progeny. So 8 varieties of progeny are produced. I will explain you in detail, don't worry. Okay. So in this cross we are going to get first parental varieties. I am going to write first parental varieties. Okay. So this is the first parental variety, that is parental mutant variety. So next one is parental wild variety. So due to first recombination, SC EC plus CV is going to be produced. So SC EC plus CV plus is produced. So this is the first recombinant variety. Next one is, so SC plus EC and CV is going to be produced. Let's see. EC plus, sorry, SC plus EC CV. So this is also a recombinantal variety occur by a crossing over, produced by a crossing over in between SC and EC location. Okay, so let's see the next variety. So second, according to the second crossing over. Okay, so you can see the second crossing over. So the variety is produced is SC EC CV plus. Okay, I am going to write SC EC CV plus. Okay, so this is also a recombinational variety. So let's see the next variety, SC plus EC plus CV. I am going to write a sixth variety, that is SC plus EC plus CV. Okay, so now we are going to see the double crossover products. Okay, so SC EC plus CV, that is the first variety. Second one is SC plus EC CV. So I am going to write, okay. So these two are the double crossover products. So okay, EC. Okay, these two are the double crossover products. So this is the single crossover recombinant. This is also single crossover recombinant. This is also single crossover recombinant. This is also single crossover recombinant. But should remember, these two are the double crossover recombinants. These two are produced due to double crossovers. Okay, these two are the double crossover products. Generally, we know that parental varieties are produced more. Here also, so first variety, that is mutant variety, completely mutant variety, produced 1158 individuals. Second variety 1455 individuals. So third variety 163. Next 130 individuals, and then 192 individuals, 148 individuals. Last two are double crossover products. So they are produced very limited number. Why? I will tell you later. Okay, so they are produced single individuals are produced. Totally, so the progeny will be produced are 3248. Okay. So here you need to understand one thing, that is, so if you observe rare recombinants, these two are the rare recombinants. So you will know which is shifted in between. Okay, So here EC is shifted from here to here, EC is shifted. So in the rare mutants, which gene is shifted, by knowing that you will understand what is the gene sequence. So from this we can assume the gene order. So the possible gene order is SC, EC, CV. So this is the possible gene order on X chromosome. Okay. So now we are going to see the distance between these three genes. That is nothing but 3-point test cross. We are going to see it. Okay. So now we are going to calculate the distance between genes. So let's see the distance between SC and EC. Okay. So you need to know, you have to see where the SC EC genes are get exchanged. Yes, in the first case and in the third case. In the first case they are exchanged, and in the third case too they are exchanged. Okay. So SC and EC. Okay, SC and EC in the first case and third case. Okay. So that's why this we should consider 3, 4 and 7, 8 products. So in third products are produced 163 plus 130 plus 1 plus 1. Okay. So that is equal to 295 by 3248 into 100. So the answer is 9.08 centimorgan or map units. Okay. Now we are going to calculate the distance between EC and CV genes. So fifth products. So let's see. And the sixth one, seventh and eighth products are having the crossing over in between EC and CV genes. Okay. That's why we are going to calculate this by 192 plus 148 plus 1 plus 1. That is equal to, so that is equal to 492 by 3248 total progeny into 100. Okay. So the answer is 10.5 centimorgans. Okay. We already know the possible gene order, that is SC, EC, CV. We are already calculated the distance between SC and EC and also EC and CV. So by adding these two distances we can calculate the distance between SC and CV. So let's see the calculation. SC, EC, CV. So the distance between SC and EC is 9.1. So the distance between EC and CV is 10.5. By adding these two we get the distance between SC and CV, that is 19.6 centimorgans. 19.6 centimorgans. Okay. So we are going to calculate the frequency of parental varieties or non-recombinants. So to this the equation is a number of parental varieties produced in the test cross by total progeny of test cross into 100. So by substituting the values we can get the frequency of parental combinations. So parental combinations are produced 1158 plus 1455. By total progeny is 3248 into 100. So we can get the value, that is 80.44. So this is the frequency of parental combinations. Now we are going to the frequency of recombinants by single crossover. Okay. So let's see. So single crossover products, they are 163 plus 130 plus 192 plus 148. They are the single crossover products by total progeny 3248 into 100. So we can get the value. So that is 19.48. So in the same way we are going to get the double crossover products. So the frequency of double crossover products we are going to get, they only produced to 1 plus 1 by 3248. So these are the double crossover frequency. So into 100. So simply we can get the value, that is 0.061. Okay. Now we are going to calculate the distance between these genes. So we can calculate them by seeing the average number of crossing overs. Okay, for this the average number of crossing overs in parental variety, number of crossing overs occur in parental progeny into frequency of parental progeny plus the number of crossing overs occur in recombinants into frequency of single crossing over progeny plus number of double crossovers products into frequency of double crossover products. So by substituting these values we are going to get the distance between these three genes. So in the parental varieties the crossing over is 0, number of crossing over is 0 into, so the frequency of parental varieties we got previously 80.4 plus single crossover, the number of crossing over, single crossing over into the frequency of crossing over, single crossing over products is 19.5 plus for double crossovers the crossing over number is 2. For this the crossing over frequency is 0.061. Okay, so this is becoming 0 plus 19.5 plus 0.122. Okay, so the answer is 19.6 centimorgans or MAP units. So this is the answer. Here you have to know one thing, that is the frequency of double crossovers is very less, that is 0.061. Why? Because it is very low or less frequency. Why? Because first crossover, I have already told you in previous class, first crossing over that inhibiting the second crossing over, it is called as interference. If the interference is 100%, then there is no double crossover chance. There is no chances for double crossing over. Okay, this is about 3-point test class. Okay.